Awesome. So let's get started. Well, thanks very much for including me in the symposium. It's really wonderful to be here, a theme I care very deeply about. So as was just mentioned, our lab is extremely interested in trying to bring big data and advanced technologies into trying to improve healthcare, which I think is broken, but I won't get into that in all the ways it's ever broken. I think just to remind people, your health is influenced by many things. You've heard themes on this today. Your genome certainly is one important part, but all these other things around lifestyle, meaning exercise, food, stress, as well as exposures, all impact your health. We know this qualitatively, but we're trying to, as other groups are, trying to define this quantitatively so we can really understand the relationships between these various parameters and impact health with the ultimate goal of improving healthcare. So as many of you may know, a number of years ago, it's actually pretty close to 13 now, we started profiling people with big data technology. Started with the omics technology, sequencing people's genomes, and later other sorts of omics that have been covered today already, including microbiome and immunome kinds of assays. This is on top of deep clinical tests and even advanced tests around stress echocardiograms, glucose controllers, and then I will spend some time on wearables. So it started on me, and then roughly around 9.5, 10 years ago, we expanded to 109 people that we've been profiling for quite some time. Some are starting to drop off, but we're adding new ones. And so one aspect of this is to collect very, very deep data around folks, and the other aspect is to do it longitudinally. So just arbitrarily, we started sampling people every 3 months while they're healthy. And then actually many more samplings when people first got ill, with the idea of what does it mean to be healthy? What does that profile look like? People don't really study health, at least at that time, not very much. And then what happens at the earliest times of illness, and from the clinical side, which I think was a theme you just heard about, can we bring these advanced technologies to be able to better manage people's health? And with regards to that last point, just from the first 3.5 years of profiling this smallish group of people, which was intended as a pilot study, we had 49 major health discoveries. Now, it is an older cohort, 53.4. So the probability of finding something go off is a bit higher, I suppose, than if they were 20-year-olds. But nonetheless, although some of these folks did learn something pretty important while they were young. And it wound up spanning a wide range of areas: hem-oncology, cardiovascular disease, metabolic disease. These things are all found pre-symptomatically. And so the way we like to think about it is that if your health is a 1,000-piece jigsaw puzzle, the way we practice medicine today, you get about 8 of those pieces, and we're trying to get 800, get a much, much clearer picture of your health, if you will. And it turned out that as we did this profiling, some of them were a big deal, like here's lymphoma, some of these pre-cancers you just heard a little bit about. They can convert into aggressive cancers. 2 people with serious heart issues. And again, some of the most important concepts here was that it was all found pre-symptomatically, and no one technology did it. Sometimes it was genome sequencing, which picked up a mutation, for example, here. Sometimes it's imaging. In this case, it was actually multiple. It was imaging plus several biomarkers, and that was very common. We found this quite a bit. And so just to dig a little bit deeper, the genome sequencing actually was quite powerful. Something like the first 13 people wound up learning something important. 12 with what are called Mendelian mutations. You heard this touched on, I think, a little bit this meeting already. These are single gene mutations that put people at risk. The classic is BRCA. If you're a woman and have a BRCA mutation, you're at high risk for breast and ovarian cancer. And it turns out 3 of the individuals here did learn something pretty important. This guy, young guy, actually had a mutation here, and his father died of a heart attack. It's a mutation, a cardiomyopathy gene. His father died of a heart attack, had one in the 50s, and sure enough, he's got a heart defect on medication now. This individual actually had a mutation. This gene, also pretty young, did a whole body MRI follow-up. They're at risk for certain cancers, and sure enough, they had early thyroid cancer. And then 9 individuals coming into the study were thought to be type 2 diabetic. One of them turns out to be a mutation in this gene. They're diabetic, which is very different, and the way you treat it's different. So 3 people learned something that was immediately actionable. Well, they're all actionable, sorry, take that back, but they learned something that was really important. Now, with the theme that was, I think, presented a little bit throughout this conference, This is just these Mendelian diseases, single gene mutations that put you at risk. But you may know that most diseases are complex disease. And in general, that's thought to be due, rather than single genes that cause a big problem, these are really thought to be due to common variants, as they're called, of low effect. And so actually these are thought to cause problems because if you inherit a lot of the bad common variants, you actually get at very high risk. And people set up what are called polygenic risk scores to follow this. And so we like to think of this, if you're unfortunate, you get all the bad ones, you're high risk. And by the way, these polygenic risk scores, they do work for the top few percent, say 2 to 3%, but they really don't work for most people. They work for me and my type 2 diabetes, but it really didn't work. It doesn't work most of the time. There are mathematical reasons for that. So we think of this as, again, single gene mutation, like knocking you out with the sledgehammer, and this is sort of death by 1,000 cuts. All right, so we've come up with new ways. I won't have time to get into it, but I will advertise some of our papers that did this, of using machine learning, because I think it fits with the theme of the conference. We can actually now, we've come up with whole new ways of analyzing genomes to get at the genetic basis of disease. And it involves using machine learning. And we did this initially for abdominal aortic aneurysm, because this is a case that's typically only discovered when people's aorta bursts and they die. There were 3 known genes when we did this. We actually, in this case, we sequenced roughly 400 people, 268 cases, 133 controls. And then we looked for an enrichment of rare pathogenic genes that had rare pathogenic variants in them in their coding regions relative to controls. And it worked incredibly well. We wound up getting 60 genes, and it's all the sort of genes that you could expect, set up a predictive model. Then we're kind of encouraged, so we decided to try a different version of the machine learning, this time looking at common variants. And again, it gets complicated, so I won't get you into all this, but basically what you can do is map out the open chromatin regions in motor neurons, which is where ALS, so Lou Gehrig's disease, is thought to occur. And then you can do machine learning, once again, in those open chromatin regions, looking for an enrichment of GWAS signals. So we took a large GWAS study, had about 13,000 cases, 24,000 controls. And that same GWAS have been analyzed that had found 7 genes, and we found 700 genes. And it's, again, we validate this by lots of means, additional cohorts, all kinds of downstream analyses. And then finally, we looked at severe COVID, same thing. We could look at open chromatin regions for all 19 cell types in lungs, including immune cells. And by looking for enrichment of GWAS signals with these new machine learning methods, went from 47, that was when it was pretty advanced. This is like 1.4 million controls, 5,000 cases. We found over 1,300 or so genes involved in this. And we could even identify the cell types responsible for severe COVID, why young people, if you will, get COVID and wind up hospitalized or die. It's because actually they have an enrichment of signal in their natural killer T cells are generally less, their genes are less well expressed for certain key genes from the set we identified. They're lower expressed. So bottom line is, a message I want to leave you with here is that machine learning is super powerful for actually even doing new forms of genome analysis. And we don't have yet predictive scores for these two, but we do for this. We'll get there for this. All right, genome sequencing was powerful. Other technologies are also powerful. Imaging picked up the early lymphoma. This person had a large spleen and also had elevated certain blood markers. This is one of the precancers I mentioned, MGUS. There's another one, smoldering myeloma. This individual had way too much IgM compared to everyone else. There's 1,000 samples here. His 2 samples were just way, way higher than the other 998. And sure enough, follow-up tests said he had too many IgM producing cells. It puts him at high risk for cancer. People have plaques in their arteries, 6 of them did. I'm one of the 6, and mine looks like this. Not huge, but they're there. And the recommendation is increase your statins or other things. I actually took recently a PCSK9 inhibitor. Boy, does that drive your LDL way, way down. It's great. And yeah, what can I say? It's my plaques are shrinking. They actually did it just by increasing statins. number of people several people were diabetic going into the study. We're very interested in type 2 diabetes. I mentioned I am type 2 diabetic. And once you know it, we had 9 individuals who thought they were diabetic coming in the study, and 2 additional ones were diabetic who didn't know it. And a lot of people are pre-diabetic and didn't know it. This is very, very common, actually. 9 out of 10 pre-diabetics, including a lot of you listening to this call, you don't know it. So we think it's important to know so you can push off your diabetes. Anyway, we were very curious, how do people become diabetic? And it turns out 9 additional individuals became diabetic during this study, and others had diabetic range values, and others became pre-diabetic. Again, when you profile people, this is just for the first 4 years now. And so it turns out of the 9 who clinically became diagnosed as diabetic, 7 of them just gradually increased. So here's the fasting glucose, here's hemoglobin A1C, both indicators of diabetes. This person gradually went up there, ultimately reached diabetic range, and so was actually treated, I believe it went on metformin and brought it down. This person got there through oral glucose tolerance test. So most people gradually became diabetic. However, 2 people basically spiked their way into diabetes as though something triggered it. I'm one of the 2. This is me. Again, my genome predicted it, actually, from the polygenic risk score. I first became diabetic after a nasty respiratory syncytial virus infection. I got it under control by changing my lifestyle, although ironically, it included cutting out sugars in my diet and running, adding running, and I got it under control. It was all fine. And then I stopped looking at my data. It's a nice controlled experiment if you think about it. Later, someone else looked at it and said, Mike, you're diabetic again. My hemoglobin A1C was 7.0 at that time, over 6.5, which is diabetic. And it turns out I'd spiked back up again. It turns out I'd stopped running right here, and I also got another viral infection. So we think, at least initially, the viral infection with genetics was triggered it here. The running may have triggered along with the viral. I started running again after I learned this, and I gradually, got it down, but it kept creeping up. Ultimately, the key for solving me was actually more data, and I think this fits with the whole theme of this conference, which is more data turned out to be very, very powerful. It turns out I'm insulin sensitive. I make insulin, so what's wrong with me? Well, my first clues were I don't dispense of glucose very well, even though when I ate a glucose strength, like a oral glucose tolerance test, I'm very, very slow at getting rid of my glucose. And it turns out I don't release insulin from the pancreas, and there are drugs for this. Rapinolide is one, and I'm a good responder. It turns out I'm a metformin non-responder, which is, it doesn't happen often, but it did happen to me. And so we call this precision diabetes. By better knowing what's wrong with me, I could do the right treatment for the right time and get my diabetes under control. I'll just go on and say we've done all kinds of fun things with this cohort. We've been able to look and show that there are actually 2 biological seasons, and they're from Northern California when their molecules peak. One is late April, early May. The other is actually in winter when certain molecules will increase. Turns out a very interesting study, in my opinion. We can also see how people age, and it turns out everybody ages a little bit differently. We think of aging like your car, where the whole thing gets older, but some parts wear out first. This is me on the left. We discovered people age differently. I'm a typical ager. My coagulation, metabolic pathways all go up. This is 4 years of data over time. This other person, though, their top pathway that goes up is a cardiac hypertrophic signaling pathway. They're a cardio ager. And it turns out we later learned this person's stage 2 hypertensive, which sort of fits with this idea of cardiovascular system's a bit off. We've gone on and classified people into ageotypes, meaning your aging pattern. So each column's a different person. People over here, their age, the 4 patterns we discovered, there are more than 4, but it's just 4 we were empowered to statistically identify. So kidney, liver, metabolic, and immune agers. And so people over here are aging in all 4 categories. This person's aging in 3 of the 4, not so much of a kidney ager. I'm somewhere in here. I'm a normal ager, except I'm not much of an immune ager. I forgot which one's me. Maybe this one, more of a kidney, liver, metabolic ager. This person's kidney, this one's kidney and metabolic, so on. So we can see how people are aging, and there are clinical markers associated with this. So we can actually make some, principal recommendations for actually reducing this prophylactically based on their trajectories. And so again, it is actionable information, kind of cool, actually. All right, now, an area that we're extremely interested in, as was introduced, is the wearables, which is a fairly new field. At the time we got involved, people were just they were just fitness trackers. And we said, well, these are pretty powerful health monitors. And they're getting quite popular in the US, 20%. In fact, I just learned this morning, somebody says now 25% of the US does wear a smartwatch or a ring or some equivalent device. And there's tons of different devices out there. But I'll say they're powerful because they're measuring you 24/7. So even the simplest watches will be making hundreds of thousands of measurements every day. They track many things. They all track heart rate variability, and they're all reasonably accurate for that. Respiration is so. Blood oxygen and blood pressure isn't necessarily accurate, but the deltas are fine, the changes, and skin temperature depends on the device. Sleep is another one. By the way, I'm wearing, if you look up, I'm wearing 4 of these smartwatches. My hearing aid is not just a hearing device. It also, I use it for hearing, but it helps measure my physiology and other activities. So these things are very, very powerful. They can be everywhere. And so we discovered early on, I want to save a little time for questions, so I'll try and go a little quickly. I hope it's not too fast. But we discovered early on you can pick up infectious diseases from a simple smartwatch, and in this case, a pulse ox. So I actually first figured out my Lyme disease because my blood oxygen was running very low. I first noticed it on aircraft, and when I landed, it didn't come back to normal. Your blood oxygen normally drops on aircraft, but this was unusually low. And as I say, it never came back to normal. I also saw my heart rate was up. This is a low resolution way of saying it, but my heart rate was running elevated. I later learned my skin temperature was up as well. I did go to a physician. I was in Norway at the time, and I was flying to Norway when I discovered this. And so he took blood, said my monocytes are up, you should take penicillin. I said, no, I need doxycycline. So we actually, he was reluctant, but to give in. And so it cleared that up right away. You take it 2 weeks. And sure enough, I was Lyme positive when I got back. And another well-controlled experiment, I'd given blood just before I left, and I was negative there. So I clearly seroconverted during this time. We went on to show that not only Lyme disease, but other respiratory illnesses you could pick up with a simple smartwatch. And actually, so both resting heart rate and skin temperature turned out to be very, very powerful ways of seeing when someone's getting ill. So for Lyme, viral infection, this turns out to be a viral infection. This one was asymptomatic, so not sure, most likely a viral infection, but had high CRP every single time I was ill. We could pick this up by high resting heart rate, high skin temperature. And we went on to write algorithms that follow your baseline and then look for the shift up in resting heart rate. And it worked great on me. This is all retrospective. Every single time for these infections, including even the asymptomatic one, we could detect it in advance of symptoms, which is the red line. This is sorry, I didn't explain this, but this is a delta plot, sort of a sliding window plot where you shift up means there's been a change from the previous baseline. And so again, it worked on me, it worked on 3 other people, one of whom got sick twice. Every single time you see this jump up in resting heart rate prior to symptoms. Skin temperature doesn't work as well because a lot of people, we think, don't put their watch on tightly. All right, so of course, we published that one now almost 6 years ago. And then the pandemic came along 2.5 years ago. So of course, we were improving our algorithms, building an infrastructure, bam, along comes like the pandemic. If you think about the way we measure illness now, it's totally flawed. It's based on oral temperature, which, it's a 300-year-old concept, and it works much of the time. It doesn't work that well for COVID because a lot of people don't get fever. So we think resting heart rate and these advanced technologies of continuous monitoring could be very powerful. So we found right away, I won't give you all the details. We quickly rolled out a study. We had one rolling. IRB said, you got to do it for COVID. So we rolled out a new one. And right away, we enrolled 5,300 people. 32 had gotten COVID while wearing a Fitbit. And this is it's our very first actually subject. And this is resting heart rate plotted as a standard deviation plot. And what you'll see is here's diagnosis day, here's symptom day. This person's heart rate jumped up 9.5 days prior to symptom onset and was presumably spreading the disease without knowing it. And here's our real-time detection method. I'll tell you in a minute. Again, all retrospective. So from these first 32, we showed that 80% of the people, we could detect COVID at or before symptoms, and the median was 4 days prior to symptom onset. So just jumping ahead, we wrote a real-time detection algorithm that actually follows your baseline. It looks for a jump up in resting heart rate, a little bit of sleep, a little bit of activity data in the algorithm. We've written on the lightweight one so we can scale this for millions and millions of people. So I want you all to sign up innovations.stanford.edu/wearables. Please join our study. And it works. I mean, 80% time. It's not perfect. You'll see the flaws in a minute, but it's not too bad. So basically you click on this, see where you're green day. That means you don't have an abnormal signal. Red day means you have something shifted up. And so here's one of our first cases again, red alert, red alert, red alert, and then got symptoms that day and diagnosed the next day. So it works. Again, this time it's about 3 days prior to symptom onset because of the real-time detection. And it works on asymptomatic cases. It works for Apple and Fitbit. I'm sure it works for all of them. It's just that we have more data for those too. Here's an asymptomatic case of, this is when they're diagnosed. And you see they had red alerts running for about 2 weeks before that. It's not specific for COVID. Other stressors will trigger it. This is workplace stress is number one. Intense exercise, not, if you run a marathon, your heart rate will be up for a few days and that'll trigger it. So other things do it, including other infections. We even picked up a mycoplasma infection, but other viral infections will trigger these red alerts. It did work for me, interestingly enough, when I was due to fly to New York City. I'm mostly green. I had some stress days there not going to get into, but when I was due to fly to New York City, I woke up that morning and I was a little bit congested. So I did an antigen test and I was negative. So I then checked my watch and sure enough, I'm positive. I'm a red alert. So what I do, I got on the plane anyway. Bad move because I got there. In order to go to the meeting, you have to get tested. And sure enough, I'm bright positive. So, and so I wound up having to quarantine a whole week in a tiny little room in New York City, very small room, kind of not fun. All because I listened to my antigen test and not my smartwatch. So what this means is that this test, the smartwatch is more sensitive than an antigen test. 2 beats per minute, you'll pick up a positive signal with our test. But it's not a specific, we don't know it's COVID, of course. We know it's just a stressor. So we need to collect other kinds of data to differentiate this better. I won't have time for this, but we can pick up people with long COVID because they have elevated resting heart rate. And again, I think this is just the tip of the iceberg because what we're finding is that you can grab features from a smartwatch and using clinical data make predictive models. And sure enough, this is some work again from some machine learning Jesse Dunn had done that these hemoglobin or red blood cell count, hemoglobin, these are signs of anemia or dehydration. You actually can get a signal out of your smartwatch, even hemoglobin A1C and fasting glucose, you can as well. So we think these are going to be measures. They're not clinical grade measurements, but they're good enough to tell you something's off, you're a bit shifted. I'll just summarize quickly. We've gone on. We do a ton with glucose monitoring as well. We put these, I think our stick is we put them on normal and pre-diabetic people. We're very, very surprised to see quite a few of them were spiking away. Some were spiking just as bad as diabetics. And so we classified people in the glucotypes based on their glucose level, low, medium, or high. And then we went on to discover, as at Aaron Siegel, that different people spiked to different foods. Some people spiked to bread, some to bananas, some to pasta. Different people, it's probably, in part, it's their microbiome, as Aaron Siegel showed. But there's other factors as well. So we think it's pretty important to know what's spiking you, and these glucose monitors are very powerful for that. So we've gone on to build a dashboard that actually collects all this wearable clinical omics data, puts it in. It's an open source dashboard, so you can display your work and such. And so this is where I see us going. I see us going into a world where genome sequencing will actually become routine, and ideally before birth. You may know there's new technologies to drive this price way down. Along with omics and wearable data, and certainly other omics mentioned in this meeting are all powerful. And I think we're going to better manage people's health. Now, I will say we're actually trying to expand this concept we just did on profiling to make it more global. And so with the Human Proteome Project, part of HUPO, we actually have been running a project where we've been collecting samples from different people around the world at their annual meeting. And it's been kind of fun. It has some interesting logistics associated. In the end, we got data from about 315 different people. Some people gave it. all these different meetings. This was a pilot initially, and then we ran it in Taipei, Dublin, Orlando. We didn't have enough, so we added on some California folks. That's the US group. But we're doing all kinds of fun analyses here. So we can cluster people by their patterns. This is by age, if you will. These are molecules that shift, either go down with age or up with age. These are early in life, and these are, you know, ones that go up throughout life and so on. And so the reason I bring that up is that we use biocrates. We didn't have it earlier on, but we're using, since we're sponsoring this meeting, we are using it as part of HPOP, which is a lot of fun, and we're getting to see which molecules change, and we want to build predictive models around that. So there, yeah, the biocrates panel is really quite, it's actually nice for this, a very targeted assay. Lastly, I'll make a shameless plug where I've spun off a number of companies from our work. One is YOLO, shown here. And they're looking for a head of informatics, if any of you would be interested in that. Here's my amazing team. I don't have time to go through. I'll just leave it at that. Clinical collaborator, Trace McLaughlin, microbiome collaborator, George Weinstock. Amazing groups of people doing analysis with the machine learning and AI. And here's our wearable team, pretty large team at one point. So, all right. And I don't know if I have time or not, Catherine. I'll leave it to you.